Hi, my name is Anne Pringle, and I'm a professor of botany and bacteriology at the University of Wisconsin-Madison. And today I'm going to talk to you about reverse ecology, which is an approach that you may not have thought of if you're an ecologist or you walk in the world and you look at things and how they grow in the environment. But I'm going to try to convince you that this is an interesting way to think about organisms that we otherwise have a hard time understanding. And I'm particularly going to focus on one of my favorite groups of fungi, the Amanita. And what you're looking at in this photograph is an Amanita muscaria. And we're going to talk a lot more about that genus as we go along. So reverse ecology uses genes and genomes to try to describe basic ecological parameters of any organism. So if you want to know, where does this fungus grow? What does it eat? What's its niche? Reverse ecology uses genes and genomes to try to answer those questions. And this approach may be particularly important if you're someone like me who studies cryptic organisms. And what I mean by cryptic organisms are those organisms that are difficult for humans to see. This image is a network of a fungus. Typically, these kinds of networks grow hidden inside of substrates, for example, soil. In this case, the network has been brought above ground by growing it in a petri dish. And you're looking at a little piece of wood and the network growing out away from the wood. We don't really have, or haven't had until recently, great tools to try to understand these organisms as they exist in their natural habitats. This is a field in North Carolina where I've done a lot of work in the past. And it's home to a really diverse community of fungi that are known as arbuscular mycorrhizal fungi. And although you can't see them in this image, if I were to take you underground and give you some kind of special goggles so you could see what was growing there, what you would see are a lot of organisms whose spores look like this, really beautiful, almost like tropical fish, except they're tiny and they live in soil and they don't swim. But they have all the colors and amazing morphologies and the amazing biology. But you can see a fish if you go scuba diving in the tropics. But these are a little bit tougher. So how do we learn about them? How do we do biology on these kinds of organisms? Well, a long time ago, or what seems like a long time ago, although actually it was only about 15 years ago, I used emerging genetic tools to try to understand the relationships between the spores of this particular species that you're looking at in this image. So I took an individual spore, and I popped it in a little tube, and I crushed it up, and I found out what the DNA sequence was of a particular gene that I was interested in. And my question was basically, are these things genetically diverse, or are they not genetically diverse? And I came up with this phylogeny that shows you the relatedness among all these different kinds of individuals. And I was really happy with that result, and I published it. It was my first scientific paper. But when I was doing that work, there was a lot that we didn't know. There were a lot of technologies that weren't available, and there just simply wasn't a lot of information. So when I compared my DNA sequences to sequences available in public databases, I came to certain kinds of conclusions about how to identify those sequences. In other words, if you submit a sequence to a public database, based on the information in that sequence, the public database spits back at you an answer, it's a chimpanzee, or it's a plant, or it's a fish. But the answers I was getting at the time were incomplete. And some years later, another group discovered that actually some of those sequences that I had generated had nothing to do with the fungus that I was interested in. They weren't from the spores themselves. They were from things that are growing on or in the spores. And they used the same sequences that I had generated, but discovered that actually, as more information became available, those sequences matched new things that were found in the public database. The end result was a prominent new branch on the fungal tree of life. Eventually, we understood that this new thing that we were seeing in the tree of life was actually a new phylum. Now, this is something that we know grows on or in spores. We don't know much more about it than that, except that other people have also discovered this new sequence in other habitats. So although we've never touched this group of organisms, we've never cultured this group of organisms, just based on these sequence data that I generated and that a lot of other people generated, a biogeography and a habitat emerge. So we can see, for example, that sequences that are new like this were discovered in Canada, in Costa Rica, in North Carolina, also my study in North Carolina, in California. So we know where on the earth we can find this particular kind of sequence that's marking this new phylum. And we also know that it comes from soils. So it comes from soils, from roots. That was also true of the study that I did. So we know something about it. And if you're interested, this is a group that's now discussed as the Archirhizomycetes. The point is that even though I've never held this thing in my hands, I know something about it, and I know something about it because of DNA sequence data. And that's really the point of reverse ecology. You can't grow it in a manipulative experiment, but through understanding, for example, how its genes are patterned across the earth, you can discover something about its niche. And it's as simple as that. And I would argue that this is something that we've been doing for the last 25 years, maybe even. It just hasn't had a name, and it hasn't had a way to formally think about what we're doing as ecologists when we use these molecular tools. So now I want to push this idea of reverse ecology, moving away from this older story about these fungi that grow in the North Carolina field, and returning to the Amanita. And this is a story that started when I lived in California, and I collected some mushrooms and discovered that those mushrooms were the deadly poisonous Amanita phalloides, or the death cap. And sometimes you'll see this poster up around parks in California to warn you that in that park there's this species, the death cap, growing. So the death cap is interesting because it kills you, and it's always fascinating to think about things that are so toxic and why they're toxic, and gosh, what happens if you touch it? Nothing, by the way. You can touch it. It's no problem. If you eat it, that's a different story. It's fascinating for that reason, but it's also fascinating because there was a rumor, an idea in the community of people who look and collect mushrooms in California that the death cap wasn't part of California originally, that it was an invasive species. And as ecologists, something that we're very interested in is invasive species. And there's certainly an idea that a habitat that is very diverse becomes less diverse when you have an invasive species arrive on the scene. So I was curious, is this thing really an invasive species? Is it really from Europe? It becomes particularly interesting because in invasion biology, we focus on plants, on animals, and on pathogens. But the death cap is not a plant or an animal, and it's not a pathogen. It's a mutualist. It's new on the landscape, potentially, but it's doing no harm. It's potentially providing a benefit because Amanita phalloides is a mycorrhizal fungus. The word mycorrhizal just means fungus, root, mycorrhizal, and you're looking at an image of a tree, and what you see in the soil is, yes, the plant, but that network is just like the network you saw earlier. It's a fungal network, and it grows in many places in the soil that the fungus cannot access, that the plant cannot access. And it brings resources to the plant in exchange for carbon compounds. That's what Amanita phalloides is. It's a mycorrhizal symbiosis. So we know or knew very little about the kinds of dynamics associated with mycorrhizal fungi that might potentially be introduced and invasive in new habitats. When I started work, the best idea available was from a guy named Herb Saylor, who in 1984 published some fantastic data based on his own observations. And the counties of California are color-coded according to where he thought this species might have been by a particular date. And if you look at this map, it certainly would suggest that this was a species that was introduced around the Bay Area by 1963 and had spread from there. But there are complications with these kinds of ideas. One is that it's just an idea. Based on his own walking in the woods, it may be true, it may not be true. There are all kinds of biases that might be a part of this work. So I wanted to know if he was right. Is Herb Saylor right? A complication in figuring out if that hypothesis is correct is that mycology, the study of fungi, is not very old in this country. Really not more than about 100 years old. And there are not very many people who study fungi formally. So there are vast numbers of unknowns when you try to answer a question like, is this species native? Was it introduced to Europe? So the first thing that I had to do to solve this mystery was really dig into the history of what people had thought about this fungus over time. And I'm going to start you in 1860. And this is an image from Barclay, is how you say it, not Berkeley, from Barclay's book of British Fungology. And I'm using it because one thing we do know is that Amanita phalloides, or the death cap, is native to Europe. So how do the Europeans think about this species? And what you see in this illustration is a beautiful image of what a European death cap looks like. And it has this green coloring on it and this very characteristic shape. If you look at a modern European specimen of phalloides, you can see it matches very well. It's kind of got this amazing green color, slightly brown on top. And other aspects of the morphology match very well. So if I click back and forth between these two images, I want you to kind of codify in your mind what the European death cap looks like. And then I'm going to move you to North America and the start of the history of the study of fungi in North America and show you that a guy named Taylor published a pamphlet, really, on poisonous fungi. And his idea of phalloides was this. And I think it's obvious now, but for lots of reasons having to do, for example, with the lack of easily available things to read, no internet, it seems obvious to us now that this is not phalloides. But I think for Taylor, who was reading, it's hard to know what sources he had access to. To him, it certainly seemed reasonable that this was a phalloides, and he labeled it as such. And he wasn't the only one. This is really what I consider to be the first major work of American mycology. Note the date, 1902. It's McIlvaine and McAdams' 1001 American Fungi. And he, like Taylor, had this conception of what phalloides is like in North America. And I love this image because you can see the elf reading his toxicology textbook as he sits among all these deadly poisonous species, including the death cap. But the point is that it seems that what North Americans were calling Amanita phalloides was different from the European conception of phalloides. But those descriptions were available to people in North America, and so even in collections, you see some confusion. Here's a specimen, a dried mushroom that has this fantastic photograph that goes with it. It's held at the New York Botanical Garden, and if you read the written description that accompanies this dried mushroom, you'll see that what this person calls the pileus, which is the cap of the mushroom, is described as cream color with brownish center. Again, cream color, not that vivid green that you saw in the original images. There's a lot of historical confusion, but it means that when Herb Saylor said this is an invasive species and this is my hypothesis, one reasonable doubt that people could and did say was, well, their collections from California dating from the early 1900s. It's mentioned in a report on fungi that dates to the 1890s. It was here. Maybe it's native. So you have a contrast between a current idea, between historical images and descriptions and a modern idea of where the heck this thing comes from. It was all a giant muddle, essentially. So, and I just want to point out that this problem is not unique to phalloides. So this is Jacob Lang, who was a European mycologist and traveled in North America. And he pointed out that when an American mycologist who knows the European species from descriptions only, from books like the one Barclay published, faces the problem whether a particular fungus that he just collected or she just collected is the one named Agaricus X by Fries, who was, in a sense, the mycological Linnaeus. He was one of the first people who put a system to our names of fungi. So when you collect a mushroom and you're trying to figure out whether it's the thing that Fries described, you will often be at your wit's end and may count on your buttons to settle the case. And I think that this quote, it will be very familiar to anyone who's ever collected a wild mushroom and tried to use field guides to figure out what the heck it is. Often you're successful, often you're like, I have no idea what it is. And it's tough. So it's a thing that mushrooms don't necessarily lend themselves so easily to identification the way that, for example, an entire plant specimen does and all the characters that are associated with that plant specimen. Also, by the way, you can dry a plant specimen and send it to Linnaeus, and he can tell you what it is. If you dry a mushroom, it loses a lot of what you might need to identify it. So it's a little tougher also to ship specimens back and forth. So what did Jacob Lang think about phalloides? Well, in 1934, he said phalloides seems to be almost unknown in America, while it is very common with us. And by us, he meant with us Europeans. Okay, so in 1962, C.W. Emmons weighed in and said, as to the moot points, whether Amanita phalloides is found in the United States, authoritative opinions can be cited for the alternative views. I hope I'm painting a picture for you of confusion, because that was exactly the state of the art. It was confused. What's going on? Actually, it doesn't seem that anyone's for sure what's going on. Isaacson Taylor in 1963, the occurrence of Amanita phalloides in North America has been disputed for some years. So this is the backdrop against which I collected my mushroom about 10 years ago and said it's a phalloides, and said, what does that mean exactly if it's a phalloides? Is it really an invasive species? But I can tell you that as of this moment, specimens with a European morphology are certainly, the European phalloides is found all around us in the Bay Area. And here's a picture of it. And here's the habitat in which it grows. And all these flags are marking various sites where during one of our collecting trips we were trying to find phalloides. And certainly by 1973 and 1977, that European phalloides was found in the US because there are authoritative publications dating from 1973 and 1977 where they really nailed it, that it has that European morphology. It doesn't, the fact that it's here by these dates and that I can find it now didn't really solve the problem of whether it was native or not. What about that reference from the 1890s that listed it? But it's a muddle. So to solve this problem, I had to take a variety of different approaches. One approach I took was to use genes to tell me whether a particular specimen was or was not phalloides. So I used a gene called the ITS. This is commonly used by people who work with fungi. And it's very different among Amanita species. So this is a slide that shows you that this section of the gene is highly variable among different species of Amanita. So depending on what I discover after I sequence this gene, I can identify a particular specimen as a particular species. It also has this highly conserved region. That's very handy. So I know that I've got the right target. I know I've in fact sequenced the ITS. So how do we use these data? Well, one of the things that I did was go back to these old specimens, get DNA, sequence this gene, and say definitively one way or the other, it is a death cap or it's not a death cap. And generally, often, these early specimens, they're not death caps. the morphology didn't match, and it turns out that there's a reason for that. In fact, it's not the death cap mushroom. And I could do this for a lot of different kinds of specimens. And in fact, what I found is that most of those early records where there was a collection were not phalloides. That doesn't really solve the problem of the literature. If someone said they found it, but they didn't leave any kind of specimen, I still have to think about and handle that literature. But it solves the problem of collections. So you can't do anything about that early literature. So instead, you have to take a different approach. And what we did is use this idea that if, in fact, there were this distinct population of the European morphology of phalloides in California, it would have its own unique genetic variants. Just like a population of humans living in Finland might have a unique set of genetic variants as opposed to a population of humans living in Siberia, for example. Over time, if populations are very far away from each other and don't interbreed, they should be distinct in some kind of signature in their DNA. So, and this had already been described for various other species of Amanita. So if I get a lot of genetic markers out, I should see this kind of signal if, in fact, phalloides is native to California. And I'm not going to show you all of those data, but I'm going to give you the punchline. It's very clear from doing the DNA work that the genotypes from California and Europe are interrelated. Those early even descriptions, even historical descriptions, aren't right. This phalloides that we see now is actually, it's not just the European morphology in quotation marks, it is the European phalloides. It is an introduced species. It is invasive in California. So quite a bit of work later, this is our current understanding of the fungus. So in California, this is where you can find the shaded gray bits are where you can find phalloides. And the arrows mark historical specimens dating from the 1930s and 1940s, where there is a specimen that I did confirm is phalloides. Interestingly, phalloides is also found on the east coast of North America, where it's been introduced and it's established as distinct populations, but it's not spreading there. It's not invasive. It's contained within parks, for example. It turns out that even though we hadn't thought about this a lot before in invasion biology, it's quite common to see these symbiotic ectomycorrhizal species introduced to different parts of the globe. So what you're looking at here is a map, and if a country is colored yellow, it means we had data for it. And if it's yellow and it has a green circle inside it, that green circle marks how many non-native species of ectomycorrhizal fungi we know exist in that country. So phalloides then becomes a case study for a pattern that seems to be happening over and over again in different countries. It's not unique. It's just that we hadn't really thought about this idea of an invasive ectomycorrhizal fungi before we started working with phalloides. So back to the idea of reverse ecology. So phalloides is fascinating, but it's a terrible model organism because I can't grow it. So it's really hard to work with it in the laboratory. In fact, it's nearly impossible to work with it in the laboratory. So after having done all this historical work and this collecting of specimens and sequencing of specimens and trying to understand patterns of genetic variation across the landscape, I wanted to know more, and I had to think about how I could know more. And for example, this was one of the questions that I really wanted to have an answer to, because when I started talking about this, people said to me, how do you know it's a mycorrhizal fungus? Maybe it's not really a mycorrhizal fungus. Maybe actually maybe it can form these associations with roots, but maybe it has a plan B and it can decompose organic carbon and live off of rotting wood, for example, in addition to being a mycorrhizal fungus. And that's an interesting problem. And I didn't have a way to think about it. And this is where reverse ecology comes into play. Because it turns out, and this was work done by Ben Wolf, you're looking at here, this list is, think about this as a list of species of Amanita. All the green Amanita are ectomycorrhizal and symbiotic. These Amanita are decomposers. That's how they live in nature. With this phylogeny in hand, Ben started to ask, what's the pattern of genes enabling decomposition across the phylogeny, and in particular, what does phalloides look like? And this is where phalloides sits on the phylogeny. So he looked at this pathway, and what's important for you to know is that this cartoon represents the structure of a dead piece of tissue. Let's call it a dead plant leaf that's fallen off the tree. So inside this leaf, there's carbon, and these enzymes, E and C and B, are different enzymes that are responsible for chewing up the dead leaf or the dead twig. And what you're seeing in the boxes here are the presence or absence of those genes in these different lineages. And I think you can see right away that there's a correlation between gene loss and the green ectomycorrhizal symbiotic species. And if you look at phalloides, it becomes very clear that it does not have the genes that would enable it to be a decomposer. It does not have a plan B. So I haven't manipulated phalloides in the laboratory, but again, just by looking at sequences of genes, presence or absence of genes, I've learned something about its ecology. So then we wanted to push the story a little bit more. And to do that, we needed to move beyond looking at individual genes and actually get some genomes. And that's what we did. We got 6 genomes, 3 from these basal lineages that are decomposers, and 3 of these ectomycorrhizal fungi. And with those genomes, we looked not just at 3 of the genes involved in decomposition, but a whole suite, dozens and dozens of genes that are involved in decomposition. And what you can see is with that more comprehensive, deeper approach, we find that our data hold up. So I should say that we don't have phalloides in here because phalloides is such a terrible model organism that we couldn't get a genome out of it. Although actually we just got one in the last couple of months, so stay tuned, that'll change. But when we did this work, we couldn't do it. But the point is that in general, things that are ectomycorrhizal don't have genes that enable them to decompose. Along the way, we discovered a few other interesting things that continue to make the genus Amanita an intriguing, emerging model for studies of the ecology of fungi in the environment. This is Tiersii. What we know about Tiersii is that it was originally collected in Texas, and that it grows in suburban lawns in the Midwest. So people quite often see this outside their houses, genuinely, just in lawns. It grows in lawns. It's a saprotrophic fungus. And it turns out that it is also a species that's expanding its range. We don't know where this fungus is native. It was first collected in Texas and described from Texas, but it's not clear if it comes from Texas. So it's hard to know whether we should call this an invasive species that's from some distant habitat, or whether we should call this a range expansion. And here are the data that illustrate how we understand that Tiersii is changing its range. In black are records of a fungus that's commonly collected. And this suggests that people were in states like Illinois, for example, and collecting this fungus, but they weren't collecting Tiersii. Perhaps it wasn't there. The one red dot is the only collection of Tiersii that exists up until 1959. As time goes by, people are still collecting the chlorophyllum, but they start to collect the Tiersii as well in places where they hadn't collected it before, which suggested that they started to find it in places where they hadn't before. And again, there's a crucial role played by people who just go out and collect mushrooms, not because they're academic scientists, but because they're curious about mushrooms. Because like Herb Saylor with the phalloides story, there were people early on who said to me and to others, this thing is moving its range. It's changing. I'm finding it here. I never found it before. I've walked in these woods, actually not the woods, I've walked these lawns for decades, and for the first time I'm seeing this species, and I can see it's Tiersii. So we have a really intriguing set of data within this genus that suggests a lot about how fungi are on the move, perhaps in response to changing global environments. So the takeaways from this talk are really 2. First, like plants and pathogens, mutualist fungi are invading novel ranges, often in places that are distant from native ranges. The second takeaway is that these organisms, which can be tough to manipulate in the laboratory, actually we can do a lot if we think about them slightly differently and we turn to genetic tools and perhaps especially genomes to describe their ecology in an approach that I and others are calling reverse ecology. And with that, I'd like to put up a slide that illustrates how many people have helped me along the way with this story. This is a slide I used to use in older talks when I was going very in-depth into collections and specimens and stories. And these are all the people who very gracefully offered up their stories in heaps of emails and letters and collections and otherwise to help me establish the mystery of phalloides when I was originally working with it. And I'd also like to thank my funding sources. Thank you very much.